Hi everyone, my name is Maddi Ibarbia and I'm a PhD student of BCBL, an investigation center that is located in San Sebastian, Spain. Today I'm going to explain you the poster that I've done for the CNS virtual meeting, which is called Role of Magnocellular and Parvocellular Visual Pathways in Object and Word Recognition. Visual recognition is a necessary first step for activities that we do on a daily basis, such as discriminating objects, faces, or also reading. The magnocellular and parvocellular pathways are the two major pathways of the visual system with distinct histologic and physiologic properties. The magnocellular pathway is highly sensitive to motion. That's why it's in charge of detecting spatial relationships and rapid changes. On the other hand, the parvocellular ventral stem is chromatically defined and is involved in visual resolution. It has been suggested that the magnocellular pathway is implicated in text perception and reading. Moreover, some studies found that children with dyslexia had deficits in both magnocellular and parvocellular pathways. However, to date, no studies have specifically characterized the contribution of these pathways to visual recognition of letter strings and reading processes. The present MRI study aims to characterize the contributions of magnocellular and parvocellular visual pathways to the recognition of objects and words. In order to understand the specific contributions of these main visual pathways on visual recognition, we developed objects and words that were biased to either engage the magnocellular or the parvocellular system. We also included words and objects without any manipulation, as a neutral condition that can be used as an experimental baseline. A total of 34 healthy adults participated in this study, where they had to make natural artificial judgments to the stimulus that they were presented. The functional task conformed to a 3x2 design, which was carried out in a single run. ROI analyses were conducted at the individual subject level to characterize the activation profile of regions of a priori interest for the main experimental conditions, including visual cortex regions, regions along the ventral occipitotemporal cortex, and regions in the inferior frontal gyrus. We also did functional pairwise connectivity analysis using beta series correlation method. Parvocellular biased and neutral non-biased stimuli have a higher brightness and perceived recognizability compared to magnocellular biased stimuli, which makes the recognition of the magnocellular biased stimuli harder compared to parvocellular biased and neutral non-biased stimuli. This difficulty consistently led magnocellular biased stimuli to lower accuracies and larger reaction types relative to the other conditions in the behavioral results of the present work. The whole brain contrast that we performed showed the recruitment of visual regions as well as classical regions within the language network. For the ROI analysis, basically, processing of objects elicited a stronger engagement of the visual cortex than word processing. We also observed a gradient in the visual cortex, where earlier areas were more strongly recruited for parvocellular biased stimuli, and the more peripheral regions were more engaged for the magnocellular biased ones. As we expected, the ventral occipitotemporal cortex showed a stronger engagement for word processing compared to object processing. We also observed that the engagement of the more posterior part of the ventral occipitotemporal cortex, also called POTS, differentiate between magnocellular biased and parvocellular biased stimuli, whereas the engagement of the more anterior part, which is called MOTS, was similar for both magnocellular and parvocellular stimuli. The interactions observed in all the inferior frontal gyrus regions were due to two effects. First of all, we found a gradient for the three of the regions for the processing of objects, where the neutral non-biased objects exhibited the highest activations, whereas the magnocellular objects the lowest. Parvocellular objects activation was between the two previous ones. On the other hand, for magnocellular biased stimuli, there was a stronger engagement for words relative to objects. Finally, we conducted examined associations between reading abilities and functional connectivity between these regions. This analysis revealed strong associations between reading abilities and the functional coactivation of the modes, a region that is responsive to words as language units, and IFG regions for reading both magnocellular and parvocellular biased words. In sum, our study revealed relevant functional correlates of the main visual pathways in visual recognition and reading, highlighting the differential roles of visual cortex regions, the ventrooccipitotemporal cortex, and the inferior frontal gyrus in these processes and the importance of the interplay among them. Thank you for your interest and attention.